So hi, this is the first video in a series of videos I'm making to teach people how to do cell culture. My name is Erica Bowen. I'm currently a graduate student. I just started my 4th year of my PhD, and I wanted to make videos to basically allow people to review what they've learned in training in my lab, but also help other people who are first learning how to do cell culture. I definitely needed to study things a ton when I first started and really engage with the protocols to really learn how to do like proper aseptic technique, which is keeping a environment that's not going to contaminate your cells. So all of this to say, I am just a graduate student in this lab. We have our own way of doing things. So if you learn otherwise in your lab, it happens sometimes, as long as you have good justification for why this is an okay technique or way of doing things too, that's fine. I have found throughout the years now, when I compare like what we do in our lab to other labs, we tend to be more strict with aseptic technique because, as you can see, this is our biological safety cabinet here. And we have it in the same room that we do like benchtop work. And then we have like our student offices over here. And it's fine for research purposes. In an ideal world, you would want a biological safety cabinet to be maybe in its own room, or even better in like a clean room. But we don't have that situation, and that's fine for what we need to do in our lab, right? Because of that, and because we're in like I would say, more risky space than usual, people in our lab need to have a more stricter and proper standard for aseptic technique, especially because we work with cell lines sometimes that don't allow for antibiotics or things that you add to the media to help protect against contamination. All that to say is, for your own purposes or whatever you need in your lab, you should sit down and think about, like, maybe you don't have a protocol given to you. You could take this protocol and then modify it for whatever you have or need in your space. So likely I am going to go back and make maybe an aseptic technique video in general. And some of the things I say in this video might be redundant to what I say in that video. But I'm really just going to be talking out loud through like the stuff that I'm doing. And if things come to mind, I'm going to say them out loud and include them in the video because it will be helpful, I hope. Actually, first thing is to go pee. Do not be stuck in the hood when you have to pee really bad. It sucks. So you go use the bathroom, wash your hands, come back in here, and then put on your PPE. So for doing cell culture, you need shoes, closed-toed, long pants, a lab coat, safety goggles, and gloves. So I'm gonna go put those on. An additional fourth thing might be wearing a mask. If you're sick that day and you find yourself coughing and sneezing a lot, you might want to wear a mask at the hood. Any mask will do, just something to prevent you from sneezing or coughing into the biological safety cabinet, because that wouldn't be great. Today's a super easy one. I need to aliquot. Aliquoting in theory is super easy. Maybe you need a video on how to do that, and that's okay. Yeah, so I will be aliquoting out pin strep. We need more pin strep divided into conical tubes, and the reason we do that is because you don't want to be freeze thawing stuff like this, meaning you thaw this, you use a little bit of it, put it back in the freezer, and then keep doing that from the same bottle. It could compromise the integrity of the reagent. So when we get something like this in the lab, you want to aliquot it out, meaning thaw it and then separate it into multiple conical tubes so that when you need it, you just grab a conical tube and you consume it. So I will be splitting this up into 20 15 milliliter conical tubes. This is 100 milliliters right here. And I'm going to put 5 milliliters per conical tube. The amount is kind of arbitrary, but I know that with pin strep and in our lab, we don't use a lot. So I don't want to put like a ton in a single conical tube and then be freeze thawing that single conical tube a ton of times, because people might only use 1 milliliter of that to make their media that day. Something else I forgot to mention is, in addition to not wanting to freeze thaw things like that, you also want to split up everything in separate containers for everybody to help minimize the spread of contamination if it does happen. If everybody's working out of the same bottle, it could be a problem. If something happens to that bottle, then now you've spread it across multiple students. Your lab may also want to aliquot out things that aren't frozen and just kept in the fridge, but do so to prevent the spread of contamination. So first things first is to thaw this, because it is completely frozen. It will probably take 30 minutes to thaw in the water bath at 37 degrees Celsius. So I'm gonna go ahead and put it in the water bath. Before I put it in the water bath, I like to take off this plastic wrapping on here, because I don't want water bath water to get in this wrapping and be trapped in there against the lid. I'm not opening this yet. If I open this outside the biological safety cabinet, it won't be sterile anymore. I'm just taking this off and trashing it, and then I'm gonna put this in the water bath. So here it is in there. It's set to 37 degrees Celsius. And if this were to be floating, I could put one of these weights on top of it to make sure that it's not floating in the water. Instead of sitting around and waiting for 30 minutes for that to heat up, I'm gonna go ahead and set up the biological safety cabinet for aliquoting. I'm first going to open this front slide. And I'm gonna turn on the light. And you don't wanna work in it immediately, like after opening it. You want it to allow it to reach laminar flow so that the air is flowing correctly in this biological safety cabinet, and that is indicated by this light here. So I'm gonna wait for this to turn green. Most biological safety cabinets have some sort of indicator to let you know that it's reached laminar flow. If it doesn't, you could wait, I guess maybe 5 minutes. If it does have an indicator and it doesn't turn green, there might be something wrong, and you might need to talk to your PI or boss about that. I'm also gonna open the trash can in the sharps. You may not have a sharps container. I won't need to use anything sharp today technically, but I still like to open those containers in case I do need them, because I don't want to touch the trash once I'm sterile and inside the biological safety cabinet. Alrighty, this is green now. It's telling me it's ready, and I've got my trash open. So now I'm gonna wipe down my workspace with ethanol, and this should be done every time, regardless of what you're doing in the biological safety cabinet. You have no earthly idea of what somebody could have done in here before you, unless you have your own biological safety cabinet, but I would still wipe it down every time just to help keep things clean. So I'm wiping it down with ethanol that I have available already in the hood in this bottle here. So I'm gonna spray my hands with ethanol, then reach in there to wipe down my surfaces. And I do that anytime I've exited the hood. And I do big sprays, not little baby sprays on my hand. I wanna coat with ethanol. And I'm wiping down the surface with a cheesecloth wipe. Things to keep in mind at this point too is that doing my best to not block the grate at the front of the biological safety cabinet with my arms. And then I throw that away in biohazard. Okay, now I'm gonna start adding things to the biological safety cabinet and spraying every single thing with ethanol. So I know that I need a tube rack to hold my conical tubes. I'm also grabbing conical tubes. All open bags are lab taped inside the biological safety cabinet and then placed over here if you have extras left in a bag inside crates. This might not be enough for what I need today, so I might have to get out more at some point. I'm also making sure to work in the back of the hood. I have a little bit of a rhyme and reason for placing typically clean things on the left side and then more dirty things on the right. I don't put a ton of thought into that though. It's more so that I'm right-handed, so I usually work on the right side and then reach for things with my left hand. If you're not using the whole bag and you want to get conical tubes out, you're supposed to shake the conical tubes to you and then pull them out and never reach into a bag, because when you reach into a bag, you risk this touching your skin or your dirty sleeve, and you want to avoid that. If there was still conical tubes left in this bag, you want to fold down the top, kind of like a chip bag. Pretend like you want to not make your chips go stale, and then roll it down, trying to get the air out. So it's rolled down like that, and then you would lab tape it and return it to the shelves that are likely outside of your biological safety cabinet or wherever you store those. Okay, so I've added the pipette aid. I'm adding 10 milliliter serological pipettes. I'm making sure to spray them with ethanol, just like everything else. Coat it, place it inside. I always add, like, technically if I do everything perfectly, I'm only gonna need one serological pipette to pipette up the pin strap and put it in the conical tubes. But if something happens and I need another one, I just go ahead and add extras in there. Sometimes serological pipettes can be cracked for some reason. That happened during shipping, and you want to just be able to get like another one really fast. I picked up 10 milliliter because what I'm going to do is just pipette up 10 mils and then split between 2 conical tubes at a time to fill each conical tube with 5 milliliters. Don't use the 25 milliliter serological pipette because it won't fit in the top of the 15 milliliter conical tube here, which I found out the hard way one time. Now I'm gonna respray my hands, and I'm gonna label each conical tube with the expiration date that it says on that bottle. On the top of the cap, I'm gonna write that this is pen strap. For my lab, we consume them really fast, so that's all I write on the tube. If you think that it's something that could be sitting around for a while, maybe I would also write the day that somebody aliquoted it and maybe additional information, but that's all I'm going to be doing on my things. And you want to label it with an ethanol resistant marker, not a Sharpie. If you write Sharpie on that and then you spray it with ethanol later, it's going to just wash off. I keep ethanol resistant markers in a beaker that's kind of out of frame. You may need to also spray ethanol markers and add that to your hood. Another quick thing, this is not really ideal sleeves for culturing right now. Currently my other lab coat's getting washed, but I've been culturing for long enough now where I'm very aware of my sleeves, and I'm not hitting them on anything, or really the biggest thing is that I'm making sure that these sleeves are not getting caught on the armrest and knocking the armrest out of where it's sitting here. And that's just something to think about too, if that's a situation that you might be experiencing. It takes some time to build awareness for clothing like that, but just keep in mind that you don't want to be doing that or hitting your sleeve on top of an open bottle or anything like that. Okay, so this is done heating up. So what I like to do for any larger bottles, I put down a paper towel here so that when I take this out, I place it on that so it doesn't get water all over the counter. And I dry off all bottles starting from the top really good, and then moving away from the cap with another paper towel. If I go the other direction, it's like pushing bacteria up into the crevice of the lid. And it's just good practice to have. Now I'm going to spray this with ethanol and place it in the hood. All right, so I had to actually get the bottle to see what the expiration date was because I forgot. Now I can look at it and write the expiration date on this. I'm actually going back right now and adding a little G on the cap because what I'm aliquoting is technically pin strep glutamine, and I don't want it to get confused with the pin strep without glutamine in the freezer. Writing expiration date on the side. Proper protocol would be for me to loosen all these caps to get ready to put a liquid in there. If I don't loosen the cap and I set this up in my hand, then I only have one hand to open it, and I'm not very good personally at opening conical tubes with one hand. But where I deviate is that I actually like to take the caps off when I just open all of them. I remove the cap and then I place the cap this side up onto the bench. Because if I try to just leave the cap on there halfway, I've knocked them off before when I go to pick up one at a time and add my aliquoted reagent to it. So this is just how I do it, so I'm not having lids fly everywhere. This is also another aseptic technique, is that I do my best to open the lid not facing towards my palm. So some people might have it like flipped and facing, I can't even do it anymore because of muscle memory. But I used to open like this and then place it onto the bench top. But correct aseptic technique is to open away from the palm, pinching with 2 fingers, and then place towards the back away from you. And then I sort of just drop it back down in there without touching any of the other ones that are open. And you want to be extra careful because this is the stock that other people are going to be used. You don't want to get blamed for contaminating everything. So now I'm going to open the pin strap bottle as well. I leave that lid on because it's easy to remove and put back on as I'm working. And now I'm going to prep my serological pipette onto my pipette aid and start pipetting up everything and putting it into the conical tubes. Make sure to open it on the correct side, not the side that liquid's supposed to come out of. Open it on the cotton side, and always open it like this. I know some people will like just punch through the paper, but that's technically not good technique either. I open it at the very, very tippy top because this whole part is sterile, to avoid touching the pipette itself with my gloves. And then I attach it to my pipette aid. I turn it so that I see what numbers I want to see. So on one side of serological pipettes, it's normally counting down, while another side is counting up from the bottom. I like to see the side that's counting up from the bottom. So it goes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. In my lab, I was taught to throw non-hazardous trash behind me like that. In your lab, you might have a trash can next to your hood for that kind of stuff. But in my lab, we toss it behind us, and then at the end of our time in the hood, we'll pick it up off the floor and then put it in the normal trash on the other side of the room. This is a quirky thing of our lab. Okay. Opening the lid again like the other ones, facing up. And now I'm gonna pipette up 10 milliliters and put 5 in each conical tube. I'm going slow as to not splatter the reagent. If you go kind of fast, it'll sling the liquid everywhere, and you don't want that, especially if it's a product containing animal products. And I'm not touching the top of the conical tube. I'm levitating just over it so that I can keep using the same serological pipette. We don't want to put a dirty serological pipette back into the bottle and contaminate this whole bottle. As it starts to get low, I tilt it so that I can get all the liquid into one side and be able to pipette it up. So I'm actually going to trash this serological pipette by removing manually like that, putting in the garbage, recapping the rest of this for now because I realized I should have got out more conical tubes. I'm going to recap all of these. As I'm picking them up again, not touching the opening. So I didn't use all of the conical tubes. Because of that, whatever extra ones, I'm just gonna add to our benchtop collection of them. Scribble through the labeling so that I'm not being wasteful with plastic. I try to do that wherever I can in here because we go through a lot of plastic. And now I'm done. I would put this back in the freezer. I will show cleanup process too, in case you're unfamiliar with that. If not, thanks for watching. With this empty and used bottle, I've had a little bit of bleach to it. Don't close the cap all the way and throw it away. This will be autoclaved in the biohazardous trash. If I close the cap all the way, I risk making a little pressurized bottle in the autoclave, and that's not good. All these things I put back. I never used this pipette, so I can put it back. I put the bag back there with the other stuff that's closed. I plugged the pipette aid back up. That's crucial. You don't want a dead pipette aid when you come into lab to do stuff. And even when I'm cleaning up, I'm still spraying my hands with ethanol to reach for anything in the hood to put it up. Some people get in the mindset of, oh, I'm done, but you're not done with aseptic technique until you've left the lab. I didn't use the vacuum, so I don't have to worry about that. I'm gonna wipe down the bench top now that it's empty. Everything's out. I'm gonna close it, and I'm gonna, since no one's going after me, I'm gonna turn on the UV light for extra sterilization to happen inside. This is also important to close the trash cans. If you want to be compliant, you should not leave biohazardous trash cans sitting open. And if you're a quirky lab like us and you have all your trash on the floor, you'll pick it up once the biological safety cabinet is closed and throw it away in the normal trash. So I'm also going to turn off the water bath, but then that's it. That's all you have to do. Yeah, thanks.